Happy snow day. What's that? Yeah, that's true. It was so beautiful to look out this morning. I love snow. I just absolutely love snow. I know it's a pain to travel in and so forth, but I love looking out at it. A couple of things. So one was I went through all of your exams and regraded them all. They will be available tomorrow in the biochem office at the regularly scheduled times, which I think starts tomorrow at about 9:30. And if you find that you can't make it to any of those times that are there, come see me sometime tomorrow, and I will help you to get your exam. Please don't bother them outside those hours. A couple things that I noticed on common questions that arose that students had relative to regrades on the exam was, one was that there was, drawing a blank here as I start to tell you the story. One was a lot of people commented on, well, we didn't have to memorize structures for this, but you asked us a structure. And while I did not, I did say that. I certainly said that you don't have to memorize structures. I did say that you're responsible for the chemistry. And the chemistry is inherent in that second carbonyl bond that's there. I didn't ask you to memorize the structure of palmitic acid, for example, or I didn't ask you to memorize specific structures of things, but you did have to know the chemistry. And if you go back and look at the lectures, you'll see I told you to know the chemistry. So I apologize if there was confusion on that, but that was, in fact, something that I had asked you to know. There were others that comments came up a few times that people made that, again, I certainly understand people's perspectives, but I didn't always agree. There were a few, of course, where I did agree. One was relating to, damn it, I'm drawing a blank again. Getting to be old. We didn't have to. This was relating to. Come on, Ahern. God, now I just completely lost it. I'll think about it. When it pops into my head in the middle of the lecture, I will come back and you'll see a light bulb go off, and then I'll tell you. But there was another one that was a common thing that was raised by students. Anybody have a specific one? No? All right. Okay. So that was one thing. The other is it looks like I'm getting through the material a little bit faster than I expected. So I am thinking I will certainly finish by the end of the term, which is an important consideration. But secondly, I will likely have either a review session and or an overview as part of the last lecture in here on Friday of next week, which, believe it or not, is only 5 lectures after today. So I'll keep you posted on that as I get more set up for that. As I said before, I am going to be, yes, Leah? So am I going to do a review session this week at all? I'm thinking what I'm going to do is I'm going to do a big overview, which will probably be near the end of next week. Instead of doing one this week and one next week, I'm just going to do one big one, is my plan. And then I will do a review session. Our exam in here is actually Thursday night at 6 p.m. And that's not my favorite time, as I'm sure it's not your favorite time either. But that's when our exam time is scheduled for. So that gives us more time to do review session and so forth. I kind of like to get these done as soon as possible so I can get the videos posted for you guys, and you can have more chance to think about those and so forth. So my preference would probably be to do the last of these the weekend before finals. That would be my preference, and I'll let you know more about that. But that's kind of what I'm thinking in terms of review session. Okay. Well, last time I went through the steps in translation, and I know there was a lot of material that was there. As I was writing the highlights, I was realizing that there were a lot of things that I went through. So I want to slow down a little bit with that. I go fast enough as it is. And actually step back just a little bit and remind you of some of the things that I talked about last time. So one of those was having to do with the process of elongation. And one of the confusion, actually there was something else that I wanted to bring up about that here, and I mentioned it in the highlights, but I didn't, I haven't said anything about it here. That actually related to the wobble position that I talked about last time, and a couple people asked me about this afterwards. And the wobble position, not this one, wobble position, was actually this figure right here. Notice that people said, well, how come that says I when it's actually hypoxanthine? Well, it turns out that hypoxanthine is actually the base that's part of inosine. Okay, so they don't have, when we have an odd one like that, we don't have adenine that's part of adenosine. So hypoxanthine is actually the base that's involved in inosine. So that's not a typo, but that's actually accurate. For our purposes, it's not a big deal, but since there was some confusion on it, I wanted to say a word about that. Okay. Anyway, so one of the things I didn't talk about was that the place where we draw the line for the end of initiation and the beginning of elongation is a little arbitrary. You'll see that if you look closely at the two figures that I chose, one was one that we created and one that came from Wikipedia. And they have slightly different places where initiation ends. So for our purposes here, we will say that initiation ends at the place where the first peptide bond has been made. You could just as easily say that initiation ends when the large subunit comes and sits on top of the small subunit. It's not a big deal. Okay, it's really an arbitrary line where we draw that. Okay, so having said that, I want to say a little bit about elongation and then move forward. So in the process of elongation, of course, what's happening is we are having the incoming transfer RNA that is being brought by the elongation factor TU, EF-Tu. And EF-Tu performs the functions that I described, which were making sure, first of all, that the proper base pairing is occurring between the codon and the anti-codon. And if that pairing is proper, then EF-Tu hydrolyzes its GTP and gets out of the way. And if that pairing is not proper, then the EF-Tu takes that transfer RNA that it's bound to and leaves the ribosome site and goes elsewhere, okay? So that the proper transfer RNA can diffuse into that site. Now remember, these processes are happening almost instantaneously because the net gain of translation is occurring at the rate of about 17 amino acids per second, okay? So that means that during that time, there are transfer RNAs that are not the right transfer RNAs that are diffusing in and are being taken out. And there's still, the process is happening at the rate of 17 amino acids per second. That's a really remarkable thing to think about, the complexity of this process. All right, well, after the proper transfer RNA is in the A site, and these things are all occurring in the A site, except for that very first initiator tRNA that was in the P site. After that has happened, then there's a covalent bond that joins the amino acid that's on the transfer RNA in the P site onto the amino acid that's on the transfer RNA in the A site. And that catalysis is made possible by the 23S ribosomal RNA in prokaryotic cells or the 28S ribosomal RNA in eukaryotic cells. So that forms a dipeptide in this case, as you can see, and leaves the transfer RNA that was in the P site empty. That is, it's lost its amino acid. And so then really the only thing that's left at that point is the phenomenon of translocation, which is simply the movement of the ribosome one codon down. And that causes everything to shift so that the transfer RNA that was in the P site moves to the E site so it can exit. The transfer RNA that was in the A site moves to the P site, and now the A site is open for a new transfer RNA to come in and repeat the process. And so as I mentioned at the very end of the lecture last time, that process continues iteratively with each time the protein growing by one more amino acid. I'm not sure if I mentioned it then, but I'll mention it here, and that is that the translation starts at the N terminus and proceeds in the direction of the C terminus, the carboxy terminus. So the messenger RNA is read 5' to 3'. The protein is made amino to carboxy terminus. Well, that process continues repeatedly until a stop codon appears in the A site. So a stop codon, you remember, is a UAA, a UAG, or a UGA. And there is no transfer RNA that's complementary to that. All right, so the transfer RNAs are not complementary to that. So what happens is at that point, the ribosome kind of sits there and does what we call stalling. It just stalls. There's nothing that happens. Well, that stalling is a signal for a protein complex called a release factor to bind to the A site. So the release factor recognizes that this ribosome has stalled. It binds to the A site. And it's the release factor that facilitates the dismantling of everything that we've seen so far. And there's one very cool factor about the release factor, okay? The release factor brings a single molecule into the ribosome as it binds to the A site. Any ideas what that single molecule might be? Water. A single molecule of water, because water makes that bond between the transfer RNA and the polypeptide chain unstable. Water breaks that bond down. The polypeptide chain is consequently released. And then everything else falls apart. The small subunit and the large subunit separate. The messenger RNA is released. And as a consequence, translation has stopped. Now, one of the things I didn't mention about the translation process, but I'll mention it here, is that the polypeptide chain, as it's being made, actually exits the ribosome through a little tiny tunnel. It goes out through this tunnel, and it's after it passes through that tunnel that the amino acids appear on the outside of the ribosome as translation is occurring. Okay, now that's pretty much the end of the process of translation. This summarizes in a few steps what's happening there, but that's about it. And we have made a polypeptide chain. Now, there's two other components to translation I want to talk about today. One are some of the differences in eukaryotic translation, and the other relates to the synthesis of proteins that are destined for places besides the cytoplasm. And so that's an important consideration as well. And they're actually made in a slightly modified process. Okay. So when we look at eukaryotic translation, as I said, the overall process of initiation, elongation, and termination are very similar to what we see in prokaryotic cells. There are some differences in eukaryotic translation, and they relate to the things that you see on the screen. The messenger RNAs, you note, are different. That is, they have modified 5' and 3' ends compared to prokaryotic cells. And they've also been spliced. They've been modified before they even got out to the cytoplasm to be translated. The ribosome sizes are different, as I mentioned last time, and I won't go through that again. And there are ribosomal RNA sizes that are different. And there's a new ribosomal RNA that's found in eukaryotic cells that's not found in prokaryotic cells, and that's the 5.8S ribosomal RNA. There is no Shine-Dalgarno sequence in the ribosomal RNA that's in the small subunit. That's the 18S ribosomal RNA. Now, I'll talk a little bit about sequence information that's in the messenger RNAs later, but they are not the equivalent of Shine-Dalgarno sequences. And finally, there's no FMET. That's the initiator amino acid. As I mentioned last time, the structure of the ribosomal RNA, of the eukaryotic ribosome is different, so that that internal chemical reaction that's possible in a prokaryotic cell is not possible in a eukaryotic ribosome. Okay. Well, the first thing I want to mention is that sequence conservation. So remember, we had the Shine-Dalgarno sequence in the prokaryotic cells that was about 10 nucleotides ahead of the AUG start site, and it formed base pairs with the 16S ribosomal RNA to align the start codon in the right place in the ribosome. We don't have that sequence in eukaryotic cells, but people have, Marilyn Kozak was the person who initiated this study, and so they've named this sequence a Kozak sequence. She studied thousands of eukaryotic messenger RNA sequences around the AUG. So there's the initiator AUG sequence right there, looking for some kind of a conserved sequence like the Shine-Doe-Garneau sequence. And what she came up with was the fact that there's no absolutely conserved sequence. However, there are things that appear to be fairly strongly preserved about 5 or 6 or even 9 nucleotides ahead of the start site. The most conserved of these are the sequences that you see at what here is labeled as position 8, 9, 10. It's a little misleading because this is what we would normally refer to as position 1 right here. That is, the first A of the messenger RNA would be the starting A for translation. In any event, what she found was that there's a set of sequences, and I'm going to read them off going from left to right here. There's not very strong conservation, as you can see over here. The size of conservation is shown by the size of the letter in each case. But the sequence that she's identified is a GCC, GCC, and then either an ACC or a GCC is the most commonly conserved ahead of the AUG start codon. That sequence appears to have some function. There are some genes where it's known that mutations ahead of that start sequence, ahead of that start codon, have some impact on the translation of those messages. The exact reason why that occurs or how that occurs isn't really known, but we do know that that these sequences do appear to play some role. So there's something that's recognizing those sequences, not a giant surprise, and helping the ribosome to position the start codon in the right place. Now, one of the things I mentioned last time about eukaryotic messenger RNAs is that it's not uncommon for the AUG to be the first AUG that appears in a eukaryotic messenger RNA. And so that may also provide some guidance, because if the ribosome binds, for example, to the 5' end of the messenger RNA and then starts sliding along, all it would really have to do would be looking for the first AUG and having a good place to stop. So again, that's another possible way that the AUG can be positioned properly in the eukaryotic ribosome. Okay. Questions about that? Everybody's ready for the end of the term. Okay. Okay. Let's see. What did I want to say here? This just simply schematically shows a eukaryotic messenger RNA. We know, of course, it has the 5' cap. We know that it has the 3' poly A tail that's out there. There is a coding region, and there are typically what are called 5' untranslated regions, which are sequences that don't make it into the final protein, and also 3' untranslated regions that are also sequences that don't make it into the final protein. You should remember that the AAU-AAA sequence that's shown here was the signal for polyadenylation. And so that signal remains in the final messenger RNA and also appears to play a role in the binding of some proteins. Okay. Well, I mentioned previously how the cap at the 5' end and the poly A tail and AAA sequence at the 3' end of the eukaryotic messenger RNAs play a role in eukaryotic translation. And so you can actually see that depicted here by this complex that's present on the screen. The complex involves several proteins, and if you see the letters IF up there, each one of those indicates it's an initiation factor. Okay? So you see EIF4G, you see EIF3, you see EIF34A, EIF4E, etc. Each of those initiation factors, as you can see, is involved in making this complex, and each of these factors plays the role in eukaryotic translation initiation. It appears that this circularization of the messenger RNA that we see in eukaryotic cells, as you can see here, is critical for the overall translation process. And I'll say an idea about that in a second. The last thing I wanted to point out was the small subunit right here. The 40S subunit is binding to this complex. So these proteins provide some interaction with the proteins in the 40S ribosomal RNA in order to facilitate the assembly of what will ultimately be that translation complex of the 40S and the 60S subunits. Okay, did I see a hand? Question? Okay. Now, why making a circle like that? Is there any compelling reason that should happen? And people have argued about this a little bit. One idea is the fact that the cap and the poly-A sequence at the end provide a uniqueness to messenger RNA sequences and therefore become targets for the ribosome to bind to. And that may be important because as we look at the number of RNAs that are present in cells, messenger RNAs are not the most abundant RNAs found in cells. Ribosomal RNAs are, transfer RNAs probably beat them as well. So there's an awful lot of clutter, if you think about it, present in a cell. And so having a way to identify which of the RNAs are ones that should be translated may have an important function in the process of translation. Another consideration relative to the cyclization, making the circle, is that some people feel that if we have a complex that looks like this, then if we keep it as a circle, then as translation proceeds and the ribosome goes marching along the messenger RNA, or I guess I'm going the backwards way, going along the messenger RNA like so, then what happens when it reaches the stop codon over here and the ribosome disassembles, that it's not physically very far away from another initiation site. So this might make it more efficient for the translation process to occur. So that's one other thought about that. Okay. So this figure I like. You'll see it again later this week when I talk about gene expression because it involves something called a risk complex that I'll talk about later. But I like this figure because it now gives you a little better picture of that overall complex. Here are these various initiation factor proteins that you saw before. You see the 40S subunit aligning with the start codon, as you can see right here, and the binding of the 60 to make the functional ribosome that goes along and makes that protein. You can see this amino acid sequence that is coming out of the tunnel that's found in the ribosome, and ultimately the release of that as happens when the release factors come in and cleave that bond between the transfer RNA and the polypeptide chain. Okay. So the last thing I want to show you about translation is this video that I popped up. If I haven't...
The information in the mature messenger RNA strand translated into a protein.
Yep.
Called
okay.
Net code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, one that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific
Okay, good enough there. It's that mechanical voice. I don't know if that. Every time I listen to that, it's like, oh my God! I hope that's not a real person that's talking like that. It sounds like a computer-generated voice. I hope it is. Okay, that at the very end, they start talking about what will be the next topic that I will discuss here, and that is the synthesis of proteins that have other destinations. Okay, so. That brings us to here. So, the process of translation in eukaryotic cells occurs in the cytoplasm. That is absolutely true. Okay? However, during the synthesis of polypeptide chains in the cytoplasm, there are signals that, if they're coded in the DNA, of course, and go into RNA, give a specific sequence of amino acids near the 5' end that tell the machinery of the cell this protein is not destined for the cytoplasm. So if that sequence isn't present, then the protein will be released into the cytoplasm, and it may be modified later, but basically it isn't targeted for somewhere else at that point. If that signal of amino acid sequence is present, however, there are proteins that will recognize it and then target it for completed synthesis in the endoplasmic reticulum. So let me just take you a little bit through that. This is a very simple scheme showing the messenger RNA being translated, and it shows the polypeptide chain emerging and being recognized by this signal recognition particle or signal recognition protein, the SRP. That is a cellular protein that is sole job is to look for and identify that signal sequence at the 5' end of certain, at the amino end of certain polypeptide chains. We can see the overall process for this occurring a little bit more clearly here. We see in the cytoplasm at the very top that the ribosomal complex has formed and the translation process has begun. The polypeptide chain has started emerging from that tunnel sequence that I described earlier. And in this case, it contains a signal sequence. And so the signal sequence then is a binding site for the signal recognition particle. And we can see that binding occurring right here. It has grabbed the amino terminus of that polypeptide chain and brings the entire ribosome, messenger RNA, and polypeptide chain down to the membrane of the endoplasmic reticulum. At the endoplasmic reticulum, the signal recognition particle binds to a receptor that's found in the endoplasmic reticulum's membrane, and that complex then transfers this entire ribosome machinery and so forth over to a membrane channel where the signal recognition particle is released, and the polypeptide chain then emerges through that membrane channel. So that membrane channel is simply a way for the polypeptide chain to pass through the membrane of the endoplasmic reticulum, and we can see it emerging through right here. And when the end is finally reached, there is a signal recognition sequence peptidase that cleaves the end of this structure so that the signal sequence is left in the membrane, and the signal sequence, unfortunately, is not shown embedded in the membrane here, but it actually is. And the protein, the processed protein, can then be modified or put into membranes as appropriate. So at the endoplasmic reticulum, there's a couple of things that can happen. One, and the most common of these, are modifications of the polypeptide chain. So there are things like glycosylations that we talked about last term, where long chains of carbohydrates can be attached to it. Glycosylations commonly occur in the endoplasmic reticulum. And depending upon the nature of the sequence of the polypeptide chain, parts of that chain may be embedded in the endoplasmic reticulum membrane as it's being synthesized. And so that's depicted over here. Now that's important because we have to have a way to put proteins into membranes. Remember that we saw all those different proteins involved in the process of signaling or transport were embedded as integral membrane proteins or peripheral membrane proteins in the plasma membrane of the cell. Those proteins start their journey into the membrane through the process I'm showing to you here. Okay? So this is very critical in terms of embedding this in a membrane, because what will happen is eventually the endoplasmic reticulum will bud off and make Golgi apparatus, and the Golgi apparatus can then ultimately fuse with the plasma membrane. And when it fuses with the plasma membrane, all of the proteins that are embedded in it will in fact become embedded in the plasma membrane through that fusion process. So that's a really important consideration then for how we get these proteins into membranes. Other proteins, for example, that don't make it into the, my pointer has died on me, that don't make it into the membrane of the endoplasmic reticulum will instead be soluble in the fluid portion of the endoplasmic reticulum. These proteins, if you think about them, one thing that can happen to them is when if they remain in that fluid the entire time, when the fusion of the membrane with the plasma membrane occurs, all of the internal parts will be expelled from the cell. And so that expulsion can then now release these proteins to go out and do things outside the cell. And it's also through this process that extracellular proteins are made and released. So this signal sequence that's found at the amino terminus of these proteins is very critical for ensuring that all of these processes that I'm describing to you can happen. Okay, questions? Yeah. Yes. So the ones that are embedded in the membrane will end up embedded in the plasma membrane. Okay? But those that are soluble inside of here will be released. Okay? So there are other ways they can go to different places in the cell. Okay? So it's a little bit more complicated than that. But as I said, if they stay in the fluid portion through the entire process, then when that membrane fusion occurs, they actually will be expelled. And so cells expel a variety of proteins for various reasons. So that's an important process to occur. Yes, Francesca. At what point is the signal sequence cleaved from the peptidase? That's actually a very good question. It happens fairly early in the process so that this then has the ability to be free throughout here to do its thing. Yeah. Other questions? Okay. Well, we have finally finished the process of translation. Now we sort of come back around and talk about a process relating to transcription. But as I like to point out to people, this process we're getting ready to talk about right now is much bigger than transcription. Transcription plays a role in it, as we will see. But the phenomenon of gene expression is a very important concept to understand. And as we will see, there are many things that contribute to gene expression. Very many things that contribute to gene expression. So let's start by looking at this at a simple level, and then we'll look at it at an increasingly deeper level. So at a simple level, DNA goes to RNA, goes to protein, which we're getting tired of talking about. And the critical thing that we're really interested in is how much protein of any given protein is present in a cell and when is it present. Because, as I noted, it's important that cells be able to not make too much protein to waste, and also to be able to control when proteins are available, because there may be times during the cell cycle that it may not be advantageous to have a certain protein present. I like this picture of this cat because the differences in color that you see on the fur of that cat relate actually to varying levels and varying kinds of proteins that in this case are almost being randomly made around the fur of this cat. Controlling protein levels is important, and you can see I've listed above 6 different ways in which protein levels can be controlled by cells, and most of these have sublevels as well. If we think about the process of transcription as a control mechanism, we can think about, well, the amount of messenger RNA that's made. The amount can be governed by the promoter. The amount can be governed by the binding of transcription factors that we'll talk about later. Okay? Other considerations for transcription besides amount include the timing. When is a messenger RNA being made? And that, too, can be governed by transcription factors for cells, from cells. In eukaryotic cells, splicing is an essential processing step in the transcription process. And splicing of messenger RNAs is actually a very big consideration because I've mentioned that alternative splicing can occur. And when alternative splicing occurs, that actually changes the nature of the protein that's being made. And so alternative splicing can have some effect on the composition of proteins being made. Splicing efficiency is a consideration. As things are more efficiently spliced compared to those that are less efficiently spliced, things that are less efficiently spliced don't make it out into the cytoplasm for translation very well. And so that can be a factor in determining how much of a given transcript is available for protein synthesis. Okay? Polyadenylation. Remember that the size of the poly-A tail helps determine the stability of the messenger RNA, so that's a factor in how much messenger RNA is present in a cell. And I also showed a slide earlier about the alternative polyadenylation. Where that polyadenylation occurs can affect the exons that make it into a final messenger RNA. The stability, therefore, which is a function of all or most of these things I've just talked about, are very important considerations. Messenger RNAs have lifetimes. They have what we call half-lives, how long it takes for half of a message to disappear. I've mentioned that messenger RNAs in eukaryotic cells are, for the most part, stabler than messenger RNAs in prokaryotic cells, and that's a very important consideration in terms of how much of a given protein is being made. The process of translation is also a factor. We haven't talked about it yet, but you could imagine that the efficiency of translation, that is, how fast translation starts on a given messenger RNA, how fast it proceeds along a messenger RNA, and whether or not translation even occurs on a messenger RNA are all factors in determining how much of a given protein is being made. And the last factor that I'll mention here is the stability of the protein itself. Proteins themselves have half-lives. Cells degrade proteins. Cells have ways of, first of all, recognizing damage that has occurred to proteins, and they target proteins that have been damaged to complexes within them that will then break down undesired proteins or damaged proteins in any way. So the half-life or the stability of the protein itself is a factor then in the overall levels of protein that are present in a cell. Okay, well, I want to start our discussion of gene expression then to think about some transcriptional controls. The only controls I've talked about to you so far have been those involving sigma factor in prokaryotic cells. So now I want to spend a little bit of time talking about how promoters work and how promoters are controlled. And the simplest of these systems exists in prokaryotic cells. The system I'm showing you here is probably the most studied transcriptional system in the world, and it is that of the lac promoter, which is found in E. coli. So let's imagine an E. coli cell floating around in your gut, or floating around, doesn't have to be in your gut, floating around in the environment somewhere. And it is a little bundle of efficiency. That little cell divides if food is abundant about every 20 minutes. And it functions because it is efficient. All cells are fighting a continual battle with the rest of the universe in that they are continually having to produce energy, and they want to do that efficiently, and they also want to use the resources that they have efficiently. And that's where this promoter factors in. So E. coli, for example, has a lot of genes that it doesn't need all the time. So a good example are the genes, or the proteins made from the genes of found in the lac promoter. In the lac, it's called the lac operon, I should say, the promoter, found in the lac operon. Okay. Now, question? Oh, okay, I'm sorry. All right. So, good example. So one of the proteins made from a gene in the lac operon has the ability to break down the disaccharide lactose. Okay? So, cells want to be able to break down lactose because if lactose is available, that's great. I can break that down. I get glucose. I get galactose out of it. Both of these I can use for energy, and everybody's happy. However, lactose isn't always available. So, if I am a bacterium in Kevin Ahern's gut, and Kevin Ahern doesn't drink very much milk or very many dairy products, then what happens? Well, lactose, which is milk sugar, comes from that. Most of the time, there's not a lot of lactose in my gut. And so those bacteria that are spending energy, you saw that it took a fair amount of energy, GTP energy, to translate proteins. Those cells that are making the gene that breaks down, making the protein that breaks down lactose, are wasting energy and will have a disadvantage compared to cells that aren't making that protein, right? However, if lactose becomes available, now if those cells that are making that protein can break down lactose, they have an advantage over cells that aren't making that protein. So in the ideal world, a cell is able to turn on the protein to break down lactose when lactose is present, and turns off the making of that protein when lactose is absent. And that's what the lac promoter is all about. So, the lac operon, I'll remind you that an operon is a region of DNA that codes for multiple protein sequences, and they're all under the control of a single promoter. That's the lac promoter in this case. Okay? Now I'm going to show you, there are actually 3 proteins. I'm going to show you that in just a minute. But for the moment, I want to show you just the promoter. So this is the region of DNA, the control region of DNA, that's bound by the RNA polymerase. It has 3 very important features I want you to notice. First of all, let's start in the middle. The RNA polymerase binding site is shown in yellow with a sort of an overlap here through this sort of greenish area. This is the part of the DNA where the RNA polymerase will bind to start transcription. The transcription start site is right here. To the right of that, there's another site labeled in blue and green called the repressor binding site. The repressor is a protein that recognizes this sequence and binds to it. And you'll notice that the repressor binding site overlaps with the RNA polymerase binding site. That means they can't both bind at once. Either the repressor can bind or the RNA polymerase can bind. The third site that's on this promoter is another region called the cap binding site. And cap is another protein that can bind. It's called the catabolite activating protein. You don't need to know that. You can just call it cap protein. Okay? Cap protein, I guess the P is redundant with protein, isn't it? All right. And you see it doesn't really overlap with the polymerase binding site. But it turns out that this cap actually helps the RNA polymerase to bind. So that's the structure of the promoter. Very briefly, looking at the structure of the messenger RNA, we see it looks like that. Okay? These regions that I showed you before, the promoter includes both the RNA polymerase site and the cap site, and the operator is where that repressor bound, or can bind. You see that there are 3 genes that are in this operon, something called LAC-Z, LAC-Y, and LAC-A. And they correspond to 3 proteins known as beta-galactosidase, which is the enzyme that breaks down galactose, something called permease, and something called transacetylase. I'll say very brief things about them next time, but the one we're going to focus on for the most part is the beta-galactosidase. Okay, that said, that brings me to today's song, which is one I cannot sing for the life of me, and so I have someone who is very able to do that, who recorded this song for me. I encourage you to sing along. Carboxyl, translation, just learn the steps of translation, and all the things they say about tRNA are true. Translation, to form peptide bonds in translation, the ribosomal cleft must bind to rRNA-EF-Tu. Translation. She finds the F-met's cargo. 60S lines up shine and go on. Needing translation. Okay, guys, see you on Wednesday.